Turkey. In this particular webinar, we will focus on the growth and development of the cranial, facial, and oral structures. First, we will discuss about the basic principles related to growth and development and talk about the pre- and postnatal growth of the craniofacial complex. Let's start with the basics of the growth and development. This is the first part of the webinar. Let's start with definitions. We will first try to understand how the face changes from its prenatal through childhood and adulthood. Let's define the terms growth and development. In fact, growth and development rely on the other, and they are under influence of genetic and environmental factors. Cell multiplication, differentiation, and organization occur, morphing the body in 3D. The fourth dimension is the time. Moyers defined growth as quantitative aspect of biologic development per unit of time. Most describe it as change in any morphological parameter which is measurable. Moyers describes the development as unidirectional changes in the life of an individual from its existence as a single cell to its elaboration as a multifunctional unit. According to Todd, growth is an increase in size, and development is progress towards maturity. The physical growth may be studied with measurement or experimental approaches. In the measurement approach, living animals, as well as humans, are subject to cross-sectional or repetitive sets of measurement. The experimental approach uses experiments in which growth is manipulated. Thus, this method is restricted to non-human samples only. The measurement approaches are craniometry, anthropometry, and cephalometric radiography. Craniometry involves measurement of human remains, thus providing only cross-sectional data. Anthropometry is a technique which involves measuring skeletal dimensions through soft tissue landmarks on living individuals. Cephalometric measurements have become a milestone from the invention of cephalostats by Broadbent. This technique combines the advantages of craniometry and anthropometry since it provides the precise bony tissue measurements without harming the subjects. The experimental approach includes vital staining, autoradiography, radioisotopes, and implant radiography. But, as previously mentioned, these methods are applied to non-human subjects only. Either gathered with measurement or experimental approaches, the data is subject to statistical analysis to draw a conclusion. The studies are classified under two categories, longitudinal or cross-sectional. Longitudinal studies involve gathering data of a group of defined individuals over a varying period of times. Cross-sectional studies analyze data from a population at a specific point of time. There are three important features related to the growth. The growth pattern refers to the proportionality of the given subject at a point of time in relation to its various size. The diagrammatic representation of the cephalocaudal growth pattern is a good illustration for the proportional changes that occur through aging. Note the relative increase in the caudal portion's growth relative to the cephalic parts with increasing age. During the first trimester of the intrauterine life, the head portion takes about the half of the whole body, while this ratio decreases to 1/12 with the body part growing faster. The second important concept is the growth variability. The growth variability indicates the degree of difference between two growing individuals. Since there is always variability among individuals, growth charts are made to compare the growth of a given children relative to standard values. The growth timing is important since it's directly affecting the results of the orthodontic treatment, especially the orthopedic applications. All the individuals are unique and don't grow at the same rate and at the same time. One should also know that the growth doesn't show uniform progress. The body parts don't enlarge at the same rate and same increment per year. The growth is especially very rapid during intrauterine life. Considering the whole postnatal growth period, there are three main sports periods: infantile, mixed dentition, and prepubertal. Growth and Development, Part 2. The second part of this webinar will include terminology related to growth, growth movements, characteristics of bone growth at cellular base, and growth control theories. What are growth site and growth center? The term growth site was first described by Bohm as perostal or sutural bone formation area responding to environmental stimuli with adaptive resorption. In other words, growth sites serve as a location in the bone where the actual bone growth occurs. Growth sites are dependent on the growth centers for growth. Growth center is an area in the bone that controls the overall growth under genetic factors. All growth centers can be growth sites, but not all growth sites can be growth centers. Bohm described the growth center as an area in which endochondral ossification occurs with huge growth contribution in the bone mass. Some years later, Koski revised the definition as an area in which endochondral ossification is serving for a sufficient length of time, resulting in huge growth contribution, increasing the bone mass. Remodeling is defined as differential growth activity involving deposition and resorption. There are primarily two types of growth movement. The cortical drift is the movement towards the depository surface by a combination of resorption and deposition. Displacement is the movement of the whole bone as a unit. There are two types of displacement: primary and secondary. Primary displacement is the displacement of the bone related to its own growth. Secondary displacement is displacement of the bone related to the growth of the adjacent bones. The bone grows with two methods based on the differentiation of the mesenchyme tissue: intramembranous or endochondral ossification. The intramembranous ossification is the transformation of the mesenchymal connective tissue with direct classification to osseous tissues. Endochondral ossification is the conversion of the hyaline cartilage prototypes into bone. The face of the infant is normally wide and short. With continued growth, the middle portion of the face grows more rapidly and the adult characteristics of the face become more apparent. The difference between child and adult faces are related to growing various parts at different rates at different times. So, we can ask the question, what controls the growth? Several researchers tried to answer this question. The general consensus is that the growth is under control of genetic and environmental factors. There are two important principles developed by Dr. Donald Enlow. Enlow's V principle is related to the bones of the craniofacial skeleton, which have a V-shaped configuration. According to the V principle, bone resorption happens on the other side of the V, and bone deposition happens on the inner side. Enlow's counterpart principle states that the growth of one bone in the craniofacial area relates to the other bones in the same region. Therefore, each bone and its counterpart bone both grow to a certain extent to maintain the balanced growth. An example is the growth of maxilla corresponding to the growth of mandible. This was the second part of this webinar. Thank you. Growth and Development, Part 3. In this part of the webinar, we will discuss about the bone cells and bone formation in detail. Bones are providing rigidity, shape, and serve as a frame for muscles to attach. They also produce blood cells. They protect our vital organs. Their composition changes through life. An adult has approximately 206 bones in his body, but newborns possess more than pieces of 300 bones, providing them enough flexibility to pass through the birth canal. Teeth are attached to the jaw bones with periodontal ligament fibers. The tooth movement is closely related to the bone metabolism. As mentioned previously, the bone's composition changes through life. Bones are composed of organic part, water, and inorganic part. The organic part consists of bone cells, and the inorganic part is also called the extracellular matrix. Bone formation and shape transformation is based on the balanced apposition and resorption process. The osteoblasts and the osteoclasts are the main cells responsible of apposition and resorption. Osteoblasts are cells with a single nucleus that synthesize bone. However, in the process of bone formation, osteoblasts function in groups of connected cells. Osteoblasts are specialized, terminally differentiated to products of mesenchymal stem cells. They synthesize a dense collagen and specialized bone proteins, which compose the organic matrix of the bone. Osteoblasts also produce hydroxyapatite that is deposited into the organic matrix, forming a strong and dense mineralized tissue. An osteoclast is a type of bone cell that breaks down bone tissue. This function is crucial in the maintenance, repair, and remodeling. It is a large multinucleated cell derived from the hemopoietic lineage. The osteoclast digests the composite of hydrated protein and minerals by secreting acid and collagenase, a process known as bone resorption. This process also helps to regulate the level of blood calcium. Osteocytes are simply osteoblasts trapped in the matrix that they secrete. They occupy small chambers in the bone called lacuna. They are networked to each other via long cytoplasmic extensions. These extensions communicate via long tiny channels called canaliculi. Osteocytes have reduced synthetic activity and are not capable of mitotic division. They have an average life of 25 years. Here is a schematic representation of the osteoblasts and osteoclasts formation. It should be also noted that osteoblasts transform to osteocytes, which are the proper bone cells. If needed, osteocytes may be retransformed to osteoblasts to secrete bone again. At cellular base, growth may occur in two ways. Interstitial growth consists of increase of the cellular size, increase in cell number, or the increase in extracellular matrix secretion. Cartilages and soft tissue show interstitial growth. The interstitial growth may continue till the extracellular matrix gets mineralized. Appositional growth occurs when the new bone tissue is deposited on the surface of the bone. Interstitial growth is the lengthening of the bone resulting from the growth of cartilage and its replacement with bone tissue. A person grows taller because of interstitial growth. This growth occurs at the epiphyseal plate and continues until the person reaches teenage years. In order to accommodate for increases in length, bone also needs to increase in thickness. This type of growth, called appositional growth, happens when osteoblasts in the periosteum deposit new bone matrix layers onto already formed layers of the outer surface of the bone. The clinical projection of the two cellular bone growth may be summarized as that the interstitial growth is lengthening and the appositional growth is thickening bone. In the third part of this webinar, we discussed about the bone cells and the bone formation in detail. In the next part, we will explain the prenatal growth of the craniofacial complex. Growth and Development, Part 4. In the fourth part of the webinar, we will discuss about the prenatal growth of the maxilla and mandible. The prenatal period of life may be divided into three: the period of ovum, the period of embryo, and the period of fetus. During the ovum period, the ovule inserts in the inner surface of the uterus. After fecundation, the ovule takes the name zygote, and at the end of the second week, its length is only 1.5 mm. During the period of embryo, the frame of all the organs and tissues are formed. This period is also called the organogenesis phase. cells continue to multiply and differentiate and organize. Disturbances during the organogenesis phase may lead to irreversible organopathies or malformations, like cleft lip palate, teeth absence of malformations. The factors affecting the organogenesis are genetics, iatrogenic factors, intoxications, viruses, malnutrition, and alcohol addiction. Following the formation of the organ frames during organogenesis starts the morphogenesis phase. The disturbances during this period will result with morphopathies, also called deformities. Embryo development begins with the sperm fertilizing an egg to become a zygote, which undergoes many cleavages to develop into a ball of cells called a morula. The early embryo becomes a blastula that precedes the formation of the gastrula, in which the germ layers are present. The embryonic disc becomes a three-layer unit: epidermis, endodermis, and mesodermis. These layers will form from different parts of the body. Following gastrulation, cells in the body are either organized into sheets of connected cells as in epithelia, or as a mesh of isolated cells such as mesenchyme. The fourth week after fecundation, the mesoderm covering the brain proliferates and curves towards the caudal end. The mesoderm layer shows bud-like proliferations called branchial arches. The branchial arch's exterior is lined superficially with ectoderm, while endoderm covers their interior surfaces. There are 6 distinct pairs of branchial arches, but only 4 are seen. The 5th and the 6th ones disappear soon after their formation. The first branchial arch is called mandibular arch, and the second is called hyoid arch. Each branchial arch is innervated with a specific nerve and contains the groups of cells to be differentiated to various anatomic units. The cephalic end curving downward and the first branchial arch pair form the primitive oral cavity called stomadeum. The cephalic end curving down forms the frontonasal process. The mandibular arch, forming the lower lateral border of the stomadeum, gives off a bud called maxillary process. The lower part of the mandibular arch grows ventromedially, and that part of the arch is now called mandibular process. From the frontonasal process shows bilateral localized thickenings that are called nasal placodes. The nasal placodes will soon form the nasal pits. At the 6th week after fecundation, the maxillary and the mandibular process join to form the mouth corner. The nasolacrimal duct and the lacrimal sac are also formed. If the nasolacrimal furrow do not fuse properly, oblique facial clefts are seen. Premaxilla is formed with the fusion of the maxillary process with the medial nasal process. If a disturbance occurs during this union, lip and/or alveolar cleft occur. During the 8th week of the intrauterine life, the tissues forming the mandible make a rapid progress. As a consequence, the tongue moves downward, and the two vertical parts of the palatal process become horizontal, start to fuse with each other, and the nasal septum. If a disturbance occurs during this union, secondary palate cleft occur. The fusion of these processes occurs with mesodermization. Mesodermization starts when two units get closer, followed by the removal of the ectoderm tissue covering the process and the merge of the mesoderm layer. The ossification of the maxilla is characterized with direct ossification that starts at the 7th week of the intrauterine life. Maxilla has two ossification centers, anterior ossification center and posterior ossification center. The ossification occurs first on the vestibule, and the ossified tissues penetrate between teeth follicles. The right and left ossified parts leave a connective tissue band on the midline, the medial palatal suture. Now, let's discuss about the prenatal development of the mandible. Meckel's cartilage is a solid hyaline cartilage surrounded by fibrocellular capsule. It extends from the otic capsule to the mandible midline. There are two Meckel's cartilage pieces that do not meet at the midline. They remain separated by a thin band of mesenchyme. This cartilage only takes the role of a guide for ossification, and it is not incorporated in the mandible during ossification. It forms the auditory ossicles and the sphenomallear ligament. Additionally, the fibrocellular capsule persists as the sphenomandibular ligament. The ossification stops at the site of future lingula. By this process of growth, the original primary center of ossification produces the body of the mandible. The ramus of the mandible develops by a rapid spread of ossification backwards, making a divergence at the level of the mandibular foramen. Three accessory nuclei of cartilage make their appearance around the 3rd month of the intrauterine life, one in the condyle area, one in the coronate area, and one in the angulus of the mandible. Except the condylar cartilage that has a crucial role in the growth of the mandible, the other two secondary cartilages are completely ossified at birth. Except this area, the mandible shows intramembranous ossification. At birth, the mandible remains ununified at the anterior part. Only 1 year after birth, the mandible will be one piece bone. In the 4th part of the webinar, we discussed about the prenatal growth of the maxilla and mandible. Growth and Development, Part 5. In the 5th part of the webinar, we will discuss about the postnatal growth of the craniofacial complex. The growth of the craniofacial complex can be divided into 4 areas: the cranial vault, the cranial base, the nasomaxillary complex, and finally the mandible. In this part of the webinar, we will discuss about the postnatal growth of the nasomaxillary complex and that of the mandible. The maxilla cannot be considered as a separate bone in matter of growth since it is closely associated to the cranial base. The maxilla develops entirely by intramembranous ossification after birth since there is no cartilage replacement. The primary displacement is the displacement of the bone by its own growth. The secondary displacement of the nasomaxillary complex occurs in a downward and forward direction as a result of the cranial base growth. Growth in the maxilla occurs into two ways: sutural and surface remodeling. Maxilla is attached to the cranial structures by a number of sutures. Weinman and Seischer pointed out that these sutures are oblique or parallel to each other, allowing the forward and downward growth of the maxilla. As the growth of the soft tissue occurs, maxilla is carried downward and forward. This leads to opening of space at the sutural junctions, resulting in new bone formation. In addition to the sutural growth, massive surface remodeling occurs to cause increase in size, change in shape, and change in functional relationships. Maxilla grows in the three dimensions: in height, in width, and in length. Maxilla follows the V principle by Enlow during the postnatal growth in height. There is resorption on the nasal side and apposition on the oral side. Besides palatal remodeling, there is continued apposition of alveolar bone as the teeth erupt. Finally, the direction of the growth also increases the height of the maxilla. Postnatal growth of the maxilla in width occurs with growth in mid-palatine suture and remodeling at the lateral surfaces of the alveolar process. In the anterior-posterior direction, the forward, passive motion of the maxilla is constantly being compensated by the bone apposition in the maxillary tuberosity area and by the palatal process of both the maxillary and the palatine bones. At birth, the two small hemimandibles are unified at the symphysial suture. The temporomandibular joint is immature. The ramus size is short, the gonial angle is obtuse, and the mandibular canal is large. The two segments of the mandible join in the first year, but a trace of separation may be visible in the beginning of the second year, near the alveolar margin. The body elongates to provide space for teeth. The subdental portion gets thicker, which enables the jaw to withstand with the powerful action of the masticotory muscles. The gonial angle becomes less obtuse. The alveolar and subdental portions of the body are usually of equal depth. The ramus is almost vertical in direction, and the angle is measuring from 110 to 112. With the loss of the teeth, the alveolar process is absorbed, and the chief part of the bone is below the oblique line. The ramus is oblique in direction. The neck of the condyle is more or less bent backward. Now, time to try to understand the simultaneous progress of the mandibular growth. In the vertical and sagittal plane, the mandible is displaced away from its articulation in each clonate fossa by the growth. The condyle and ramus grow upward and backward into the space created by the displacement process. The ramus remodels as it relocates posterior-superiorly. The corpus of the mandible grows in 3 dimensions: in length, in height, and in width. The corpus growth in length occurs as the ramus is relocated posteriorly. The corpus becomes lengthened by remodeling. Growth in height of corpus depends mostly on alveolar growth and results also from remodeling process of basilar border. Following the synostosis of the symphysial area, the transversal growth occurs by periosteal growth, apposition on lingual surface, and resorption on external alveolar surface. In the 5th part of the webinar, we discussed about the postnatal growth of the craniofacial complex. Thank you for watching.